Hello everyone, my name is Neha. I am a student of BSc Chemistry on the 6th semester. Today I will give my presentation on the topic coenzymes. So first of all we will begin with the coenzyme derived from pantothenic acid, that is vitamin B5, which is known as coenzyme A. So to write the structure for coenzyme A, we will first write down the structure of pentonic acid, which is as follows. So this is the structure of pentonic acid, and here this is the peptide linkage. Now this molecule is associated with beta alanine, which is as follows. Here this portion is termed as beta alanine. Now for the formation of coenzyme A, this structure, which is known as pantothenic acid, combines with cysteine to form peptide linkage. That is, we will first of all write the structure of cysteine, which is CH NH2 COOH CH2SH. Now from here a water molecule is lost, also accompanied with decarboxylation of cysteine, and then the whole unit combines with an ADP derivative. Here one water molecule get lost from this derivative of ADP and vitamin B5 molecule as follows. And this finally leads to the formation of the structure of coenzyme E, which can be drawn as hence we get the structure of coenzyme A. This coenzyme A acts as an acyl group carrier in a variety of metabolic reactions. For example, in TCA cycle, that is tricarboxylic cycle, or we can say Krebs cycle. Now we will study about the structure of coenzyme derived from niacin, that is vitamin B3. Two coenzymes are derived from lysine, which are NAD and NADP. The only difference in the structure of NAD and NADP is the presence of phosphate group in NADP. Full form of NAD is nicotinamide adenine dinucleotide, and NADP stands for nicotinamide adenine dinucleotide 2-phosphate. So first of all, we will draw the structure of NAD. First of all, we will draw the structure of ADP. This is the structure of NAD. Now similarly, we can draw the structure of NADP also, which is as follows. The only difference here is the presence of phosphate group at 2' position in the lower ribose ring. Now we will study about the coenzyme derived from lipoic acid. We will study the structure of coenzyme derived from lipoic acid, which is as follows. And from this side, the lysine enzyme gets attached, leading to the formation of the coenzyme. It is essential for the decarboxylation of alpha ketoesters. Now we will study the coenzyme derived from vitamin B1, that is thymine. And coenzyme derived from this vitamin B1 is commonly known as TPP, which stands for thymine pyrophosphate. This is the structure of thymine pyrophosphate. This is important coenzyme in energy extraction and in catabolism of sugar and amino acid. Now we will study about the energy production in living organisms. So first of all, we will study about the first step for energy production, which is known as glycolysis. In this particular process, glucose gets converted into pyruvate in a sequential manner under the influence of different kind of enzymes. So let's get started. First of all, glucose, which is a 6 carbon compound, in the presence of glucokinase or hexokinase as an enzyme, and magnesium Mg2 positive present as a cofactor, gets converted into glucose 6 phosphate. During this reaction, one molecule of ATP gets converted into ADP plus one molecule of inorganic phosphorus. Now in the next step, glucose 6 phosphate gets converted into fructose 6 phosphate in the presence of phosphoglucose isomerase enzyme. Then, in the presence of enzyme named as phosphofructokinase, fructose 6 phosphate gets changed into fructose 1,6 diphosphate. Now in the presence of enzyme aldolase, fructose 1,6 diphosphate may opt for two ways. Either it will get converted into dihydroxy acetone phosphate, or it will get converted into glyceraldehyde 3-phosphate. Here, dihydroxy acetone phosphate gets converted into glyceraldehyde 3-phosphate in the presence of the enzyme triphosphate isomerase. Now glyceraldehyde 3-phosphate in the presence of glyceraldehyde 3-phosphate dehydrogenase phosphate dehydrogenase enzyme gets converted into 1,3-diphosphoglycerate. Now 1,3-diphosphoglycerate in the presence of enzyme known as phosphoglycerate kinase gets converted into 3-phosphoglycerate. Then this 3-phosphoglycerate, in the presence of phosphoglycerate mutase enzyme, gets converted into 2-phosphoglycerate. Which in the presence of enzyme known as enolase gets converted into phosphoenol pyruvate. Now phosphoenol pyruvate, in the presence of the enzyme known as pyruvate kinase, gets converted into pyruvate. Hence, this completes the process of glycolysis. Also, here the thing to be noted is that we have started from a 6 carbon compound, that is glucose, and at the end we get pyruvate, which is a 3 carbon compound. Hence we will get 2 moles of this. This process of glycolysis is also known as EMP pathway. Also, one special point to be noted regarding glycolysis is that it takes place in cytoplasm in absence of oxygen. That is, glycolysis is an anaerobic process no matter what, if oxygen present or not. Now, after the conversion of glucose into pyruvate, oxidative decarboxylation process takes place. In which one CO2 molecule and 2 NADH molecule are evolved, and pyruvate gets changed into acetyl coenzyme in the presence of oxygen. But what happens if there is absence of oxygen? In the absence of oxygen, fermentation takes place, which is of two types: lactic acid fermentation and alcoholic fermentation. Now, this acetyl coenzyme gets converted into citrate, which gets converted into oxaloacetate, and this again get converted into citrate, and this cycle moves on in the similar manner. This cycle is termed as Krebs cycle or tricarboxylic cycle, that is TCA cycle. Krebs cycle takes place in the matrix of mitochondria. In Krebs cycle, pyruvate in the presence of the enzyme pyruvate dehydrogenase gets converted into acetyl coenzyme. With the evolvement of one CO2 molecule. Now this acetyl coenzyme in the presence of citrate synthase gets converted into citrate. Now this citrate in the presence of enzyme aconitase gets converted into cis aconitate. Cis aconitate in the presence of aconitase enzyme gets converted into isocitrate. Now this isocitrate may undergo two pathways. Either it can get converted into oxalosuccinate or in the presence of isocitrate dehydrogenase it gets converted into alpha ketoglutarate. If isocitrate follows the second pathway in which it gets converted into oxalosuccinate, then in the presence of enzyme carboxylase, it also gets converted into alpha ketoglutarate. Now, this alpha ketoglutarate in the presence of enzyme alpha ketoglutarate dehydrogenase gets converted into succinyl coenzyme A. Now this succinyl coenzyme A in the presence of the enzyme succinyl coenzyme A synthase gets converted into succinate. This succinate in the presence of the enzyme succinate dehydrogenase gets converted into fumarate. Now this fumarate gets converted in the presence of enzyme fumarase gets converted into maltate. Maltate in the presence of maltate dehydrogenase gets converted into oxaloacetate, which again gets converted into citrate, and this process moves in this circular manner. Hence this cycle continues, and this cycle is known as Krebs cycle or tricarboxylic cycle. During this Krebs cycle, CO2 which we breathe out is produced. Now we will study about the process oxidative phosphorylation. So in this process, ATP is produced as a result of transfer of electrons from NADH or FADH2 to oxygen through electron carriers. This process, oxidative phosphorylation, takes place in mitochondria and is a major source of ATP formation in aerobic organisms. Here, this is the phospholipid bilayer. Here, this area will be represented as inner membrane space, and this lower part is known as matrix. Now this phospholipid bilayer comprises of various protein complexes, that is, we can represent it with protein complex 1, protein complex 2, 3 and so on. Here we will discuss 5 protein complex in detail. For instance, protein complex 1 is ubiquinone oxidoreductase. Protein complex 2 is succinate dehydrogenase. Protein complex number 3 is cytochrome C reductase. Protein complex number 4 is cytochrome C oxidase. And protein complex number 5 is cytochrome A and A3. Now we will discuss the process that how the molecules of NADH and FADH2 gives electron for the production of ATP. So if we take the case of NADH, it breaks down into NAD plus electron plus H positive ions. Now these two electrons given by NADH are accepted by the protein complex number 1, which is known as ubiquinone oxidoreductase, and passed on. Similarly, in the case of FADH2, it gives FAD plus 2 electrons, which are accepted by protein complex number 2 named as succinate dehydrogenase, and this process of electron transfer in the phospholipid bilayer takes place sequentially from protein complex number 1 to 2, then from complex number 2 to 3, then from 3 to 4, and then finally to the cytochrome C, A and A3, protein complex number 5. During this process, the enzyme named ATP synthase finally plays the role to produce ATP in the end of the process. Now we will study about enzymes. Enzymes are globular proteins which act as biological catalyst in some specific biological processes to make them take place with high rate under mild conditions of temperature and pressure. Now we will study some characteristics of enzymes. There are usually two types of enzymes. One are those which are purely made up of alpha amino acids, that is only possess protein part, and the second one are conjugated proteins, that is which also comprised of non-protein part, and that non-protein part is known as prosthetic group. Prosthetic groups are of two kinds. One is cofactor and another is coenzyme. Cofactor is the non-protein part, that is the metal ion attached to the protein part, that is enzyme. For example, iron in hemoglobin. And the coenzyme is the non-protein part, that is organic molecule attached to the protein part that is associated with the enzyme. Coenzymes are the non-protein part which plays the important role to activate the inactive enzymes, that is the protein part. The fastest enzyme is carbonic anhydrase. Now, characteristics of enzymes are: the molecular mass of enzyme ranges from 10 raised to the power 4 to 10 raised to the power 6 gram per mole. Second characteristic is that they are highly specific in nature. Also, they increase the rate of reaction, that is speed up the reaction. Enzymes act as biochemical catalyst. They act properly at 37 degrees Celsius temperature. Also, enzymes work properly at a particular pH. Now we will talk about the specific action of some enzymes, like in case of carbohydrates, they are decomposed or they are converted into glucose by the enzymes named as amylase, sucrase isomaltase, maltase or lactase. In the same way, proteins are converted into amino acids in the presence of enzymes pepsin, trypsin and peptidase, and oils and fats in the presence of lipase gets converted into fatty acids. Now we will talk about the mechanism of enzyme action. First type of mechanism that we will study is lock and key mechanism. In this, the first step is the binding of enzyme to the substrate forming ER complex. After that, the formation of product takes place, and in the third step the enzyme gets detached from the complex formed in the upper process. The main function of the enzyme is to lower the activation energy, as we can clearly see in this graph that in the absence of the enzyme, the intermediate is formed at higher energy value rather than the case in which the enzyme is present. Due to the presence of enzyme, the activation energy get lowers down and leads to the formation of product as shown in the graph. Now we will study about the induced fit enzyme mechanism. It can be represented by, let us represent this as an enzyme, this structure for enzyme, and now this reactant molecule tries to get attached with this enzyme, but as we know that here the active site of enzyme cannot accommodate this reactant because their shapes are different. But in some examples, it is observed that the product leads the formation. Hence, this means that here a change in shape is followed with the formation of the product by the induction of the favorable type of shape according to the reactant. Here this is enzyme reactant complex. In the next step, we get the product formed, gets detached to this from this enzyme. We get our enzyme back, which is again used by another molecule of reactant, and the same process takes place in the similar manner as explained earlier, and we get our desired yield of product. Now we will study about the applications of enzyme. Enzymes are of great importance in industries for the manufacture of wines and beer by fermentation of carbohydrates. Different enzymes are used for different purposes like Sucrose is converted into glucose and fructose in the presence of invertase. Glucose is converted into ethanol by zymase enzyme. Starch into maltose converted by diastase enzyme. Maltase is converted into maltase enzyme converts maltose into glucose. Urease enzyme is used to convert urea into ammonia and CO2, whereas pepsin enzyme plays important role in producing amino acids from proteins, and the enzyme called lactobacillus converts milk into curd. Also, some enzymes are important in curing the diseases. For example, streptokinase is used to dissolve the blood clot. It checks the heart attack. Whereas phenylalanine hydroxylase is used to avoid the disease known as phenylketonuria, which occurs due to the accumulation of compound brain damage and mental retardation is caused in this disease. Tyrosinase enzyme is used to and is essential in avoiding the disease albinism. This albinism pigment melanin is not synthesized, which results in the disease albinism, and production of medicines is an important sector in which enzymes are crucial. For example, insulin, penicillin, etc. For the production of cheese by coagulation of milk, and in the manufacture of sweet syrup from cornstarch. So these were the major applications of enzymes. Now we will study about fermentation. As explained earlier, fermentation is of two types: lactic acid fermentation and alcoholic fermentation. First of all, we will discuss about lactic acid fermentation. In this, glucose gets converted into pyruvate, which we saw earlier in the process of glycolysis. During this conversion of glucose into pyruvate, 2-ADP plus inorganic phosphorus gets converted into 2-ATP net, and 2-NAD plus gets converted into 2-NADH. Now this pyruvate gets converted into lactate, along with the conversion of 2-NADH into 2-NAD plus, which are again utilized in this step to continue the cycle. Hence, this was the lactic acid fermentation. Point to be noted is that in this lactic acid fermentation, 2 moles of pyruvate and 2 moles of lactate are formed. Also, lactic acid fermentation is of two types, that is homo and hetero. In case of homo, both molecules of pyruvate gives lactate, but in case of hetero, one molecule of pyruvate gives lactate and the another one gives alcohol, in the manner that glucose gets converted into pyruvate. This pyruvate changes into acetaldehyde. Acetaldehyde converts into ethanol. In the case of alcoholic fermentation, this process is followed with the only difference that here 2 molecules of pyruvate gives 2 molecules of acetaldehyde and hence 2 molecules of ethanol. In the same way here, when the glucose gets converted into 2 pyruvate, 2 NAD plus gets changed into 2 NADH and 2 ADP plus an organic phosphorus gives 2 ATP molecules net. And in the next process, similarly, when acetaldehyde gets converted into ethanol, 2 NADH gives 2 NAD plus, which are again utilized in the first step of the fermentation, and hence the process continues. So this was the alcoholic fermentation. Now we will study about the applications of fermentation. It is used in breweries to make wine and alcohol. It is used in bakeries for making cake and other items. It is used in making vinegar. Also is important in the tanning of leather. Ethanol made by this process of fermentation have wide application in the field of chemistry itself and other medicinal uses.